Thank you everyone for coming back to our final session of the conservation genetics across multiple species. Today we're going to be broadening our scope a little bit to discuss range-wide patterns of conservation genetics. Our first talk is by Madeline Eppley, who's a PhD candidate at Northeastern University. And Madeline works on the spatial and temporal evolution of the eastern oyster. And today she'll be sharing some insights from contemporary range-wide population structure. I apologize, they will be sharing contemporary range-wide population structure.
Cool. Thanks so much. Cool. Thank you so much to the symposium organizers for putting this together. The morning talks were great, and I'm excited to open up the afternoon session. So we're all here in part today because species are under this immediate challenge to adapt to increasingly severe and rapid environmental events that are increasing in both frequency and duration. But there's a silver lining, and it's that the non-random mortality from these selection events can facilitate rapid adaptation to these new environments and potentially result in evolutionary rescue, which is where the adaptive alleles in the population arise after a period of environmental stress and allow the population to persist under future iterations of that environmental stressor. And this dynamic can happen from standing genetic variation, from de novo mutation, or gene flow. But empirical work shows that the surest path to evolutionary rescue is through standing genetic variation in the population. So I'm going to talk about this in the context of our conservation challenge for today, which is the eastern oyster. It's a declining species of ecological, economic, and cultural importance to Canada, the United States, and Mexico. So the eastern oyster naturally ranges along the Atlantic and Gulf coasts. And as we can see, we've lost a great majority of the historic reefs for this species. Along the Atlantic coast, we've lost upwards of 90% of the historic abundance. And one reason for this decline, aside from human pressures, is co-infecting biotic stressors. So parasites are proving to be an obstacle to conservation, and we're still working to understand what the adaptive responses to these rapid biotic selection events might be. And so I'm going to focus on two parasites today. The first is dermo, which we can see in this plot of disease prevalence over time, that under climate change conditions, the parasite prevalence is intensifying. And for the second disease, MSX, while it's not intensifying, it's actually expanding. So just within the past 2 weeks here in Canada, there's been novel observances of MSX in Canadian oysters at Prince Edward Island. So these parasites are proving to be a real threat. However, we know that oysters can rapidly adapt, so evolutionary rescue to these biotic stressors may be plausible. And there's two reasons that we know that oysters can rapidly adapt. The first is the combination of life history strategies, so high fecundity and early mortality allow selection to strongly influence the populations. And secondly, from empirical work, we've seen that domestication in aquaculture or selection lines can produce rapid genomic changes under just a few generations of time. So in this plot, we can see wild oysters in blue and selection lines in yellow with runs of homozygosity on the Y. And after just a few generations of breeding and selection, or in domestication, we see increased runs of homozygosity in our selection lines, and increased heterozygosity and allelic richness remain in our wild populations. So with all of this, what do we need to know in order to conserve the diversity that exists in current wild populations of eastern oysters? I'm first going to talk about the contemporary range-wide population structure of the eastern oyster. So in my work, we've collected oysters, 20 from each of these 40 sites across the range. We have sites as far north as Nova Scotia in Canada and as far south as Laguna Madre in Texas. We collected oysters from these sites and then sequenced them with a 200K SNP chip. And here's a look at the PCA for these range-wide populations. So the first thing I want you to notice is the Gulf of Mexico populations in green. We see a pretty large genetic break between the Gulf of Mexico oysters and the rest of the Atlantic seascape, which is in orange, blue, and purple. Secondly, I want you to notice the really tightly clustered wild populations. And these two trends are consistent with published literature that's come out in the past couple of years, where they use a much smaller number of wild populations, 4 and 9, and combine that with selection line data. So we're going to look at one interesting point here, which is this pink circle. It's very tightly clustered. This is oysters that are from Apalachicola Bay in Florida, and they have separated slightly from the Gulf. And so potentially what we're seeing here, one hypothesis might be a bottleneck and drift in this smaller population. And so I'm going to talk about the conservation context of Apalachicola Bay, Florida. They experienced a complete fishery collapse of the eastern oyster in 2012, and this was due to an epizootic from the parasite dermo. So they had a year of drought. Dermo thrives under high salinity conditions, and so this drove the parasite to be in very high prevalence in the local waters, and also from cumulative decades of overfishing. And as a result, there's now a moratorium on all fishing in Apalachicola Bay during this 5-year period from 2020 to 2025. And they've implemented a large restoration budget, but so far have seen low population recovery. And so this paper, Hinton Lang et al. 2023, brings up a few ideas as to why they're not seeing population recovery. They may be missing a genomics-informed approach, failure to manage biotic stressors, and not considering the temporal trends of the data here. So what can we learn from this instance of Apalachicola Bay, Florida? We should be taking into account the historic trends of our data and also responses to biotic stressors. So I'm now going to talk about some preliminary work and plans that I have for incorporating this. And we're going to evaluate how population size and diversity are changing in the eastern oyster after a past rapid biotic selection event. And so this before data that I'm talking about is before and after a period of environmental stress. So far we've looked at the modern genetic data, which, as we're familiar with, we can use to estimate parameters like genetic diversity, inbreeding, and population size. But this before genetic data can give us really valuable information too. It can show us past changes in genetic diversity, past levels of inbreeding, and it can tell us the story of how genomic adaptation may have played out in the past to the same environmental stressors that we're seeing today. And when I say before data, I do mean historic and ancient DNA, but also just time series DNA. So this doesn't have to be samples that were collected, tens and tens of years ago. We can have samples that were collected just a few years prior, and now we've had an event, and we can use that before data. So in this system, there's been a vast data collection done in the Chesapeake Bay. And so my collaborator, Dr. Ryan Carnegie at VIMS, is overseeing this collection of eastern oyster tissues that have been collected since the 60s. And each year, systematically, they've gone through several sites in the Chesapeake Bay, 2 of which are the Rappahannock River. It's a lower salinity tributary further north in the Chesapeake Bay, and the James River, a higher salinity site further south. And they've preserved these oysters in FFPE preservation. This stands for formalin fixed and paraffin embedded, so paraffin's like a waxy material. And so all of these collections are available for genetic extraction down at VIMS. And we're going to hone in on a very specific period of time in history. So we're going to look at the Chesapeake Bay epizootic, which took place between 1999 and 2002. And this is a rapid drought-induced biotic selection event. And if that sounds familiar, it's because it was the same cause of the Apalachicola Bay decline in Florida. And this epizootic in the Chesapeake Bay caused more than 90% mortality of previously healthy oyster reefs. So this is a massive mortality event. And what I want you to notice from the plot on the left is that salinity over time. During the epizootic period, we see high salinity without abatement. So it's not going down, which kills off the dermal parasite. So the parasite is just thriving during this period of time, increasing in prevalence, the plot on the upper right, and causing mass mortality. And so my first challenge with this collection was to understand if the FFPE preservation was preserving genomic DNA in a way that would allow us to extract it and sequence it. So I took a trip down to VIMS to look into the collection and wanted to understand if the FFPE tissue prevented genomic DNA degradation and yielded enough DNA for our purposes. And so I found that while DNA does degrade over time in the FFPE, this is no surprise, we do have suitable yield for genomic sequencing, which is great news. So I started with a 20 micron slice of FFPE tissue, which is pretty thin, and I realized that was nowhere near enough, so we scaled up our extractions, and we've gotten a good amount of yield. And we can see from the study period of interest on that graph on the left that we have lower yield than samples that were extracted from tissues that were preserved that year. but it's still a viable amount. So the next steps in my method are to extract DNA from 250 study individuals, and then we're going to do low coverage whole genome sequencing. And so once we have those sequences back, we're going to do a variety of analyses. The first will be to examine the effective population size and genetic diversity from pre to post epizootic. And this is going to be a within river comparison. So we're going to compare the James to itself pre and post, and the Rappahannock River pre and post epizootic. And we're anticipating to see declines in both the effective population size and the genetic diversity post epizootic. This is going to mirror what we may have just seen in the Apalachicola Bay oysters. Secondly, we're going to look at the population differentiation between rivers, so between river comparison, and we'll compare the James to the Rappahannock pre and post. We're expecting to see population differentiation increase after the epizootic because populations are going to be smaller and reduced and more subject to drift. And again, this might mirror what we saw with the Apalachicola Bay divergence from the rest of the Gulf of Mexico and our more contemporary samples. And lastly, we're going to look and see if we can identify any loci that are associated with immune response post-epizootic. And so we may see alleles that are involved with a resistance or a tolerance approach to these diseases, and we'll do a within river comparison there as well. And so out of this work, I hope that a couple of recommendations will arise for conservation of the wild eastern oyster. The first will be to provide regional or updated regional or range-wide assessments of population structure and size. As we've seen, this system can change quickly, and so as we progress through time and experience more and more biotic stress and environmental changes, we'll want to update our assessments of current wild population diversity. The second will be to use before and after data when available. So the insights that we get from combining these types of data will help us better understand how selection events have played out in the past and will inform us moving forward. And lastly would be to consider biotic stressor dynamics. So hopefully we'll be able to find some loci that are associated with disease response. We may be able to inform selective breeding. But more importantly, potentially, is conserving wild populations that may have historically evolved disease resistance. So thank you. And I'm happy to take any questions.
Super cool system.
Thank you.
I was curious if the FFP preserved samples is the DNA fragmented into a short life?
Yes. I haven't done a thorough analysis of this yet. I've looked at the gels, and I can imagine it's somewhere between 150 and 300 base pairs, length fragments. Yeah. So compared to some ancient samples, that looks all right. I might have time for one more. Sure.
Are there populations that you've seen that have reoccurring epizootic events?
Yeah, that's a great question. So the epizootic events have really fluctuated over the past 50 years. We have multiple diseases that are all driven by different environmental stressors, so some associated with temperature, some with salinity. So as conditions are variable, we can expect epizootics to be as well. Just in the Chesapeake this past fall, there's been an MSX outbreak in epizootic that's currently ongoing. Yeah. Cool. Thank you all.
Next up, we have Rebekah Rogers, who is an associate professor and lead faculty for the UNCC Evolve Research Group at the University of North Carolina in Charlotte. Dr. Rogers' research focuses on the population ecology and evolutionary processes and how those differ in small and endangered populations.
Thanks so much. All right, I really appreciate the chance to talk in the conservation section today. So I'm a professor of bioinformatics and genomics, but at heart I'm really an evolutionary geneticist. So we know that evolutionary theory predicts that in large populations, evolutionary processes will differ from what we see in small populations. So when population sizes are extremely large, there's this nearly neutral threshold below which mutations act as though they are largely defined by evolutionary processes of genetic drift, and stochastic fluctuations will allow detrimental variation to spread through populations that would normally be weeded out in a very large population. So long as 4 times NE times S is greater than 1, selection rules the day. So if you get a beneficial mutation, it can establish a deterministic selective sweep, spread efficiently through the population, and facilitate adaptation. If you have something that's detrimental, it will be weeded out of the population relatively easily. In small populations, the nearly neutral threshold will be different, and so you get this difference in adaptive processes and what acts as though it's effectively neutral. We also know that the amount of standing genetic variation in a population also depends on the effective population sizes. So theta is equal to 4 times the effective population size times the mutation rate. So we should see greater amounts of standing genetic variation in a large population. You have more individuals to collect mutations and longer coalescent times compared to what you observe in very small populations. And this is the amount of genetic variation right now today that would be available if a population gets hit with a massive shift in the selective pressures, what's available for adaptation right now. If you don't have a mutation that you need in order to adapt to shifting selective pressures, then you have to rely on new mutations. The amount of time it takes to establish a deterministic selective sweep, to have a brand new mutation appear in the population and start to spread, also depends on NE. So we have shorter time scales to identify new mutations, and then once you get them, then they are less likely to be affected by genetic drift in a large population compared to what happens in a small population. And these theories of nearly neutral evolution are fairly well defined for single base pair mutations. But in the Rogers lab, we work on sources of genetic novelty, mutations that copy and shuffle pieces of DNA and move them around the genome. And there's a lot of theory that you can read about in Mike Lynch's beautiful textbook of why nearly neutral processes might be even more important for these types of mutations. They have different selection coefficients, different mutational dynamics compared to SNPs, and they're less well understood compared to what happens in SNPs where they accumulate in clock-like fashion. So in my lab, we use a data-driven approach to try to catch these mutations in action and see how they factor in evolutionary processes in different types of organisms. So these days, excuse me, my asthma's been bad. These days I'm an evolutionary geneticist, but this wasn't always the case. I grew up in a town called Florence, Alabama, on the banks of the Tennessee River. It's right across from Muscle Shoals, and Muscle Shoals is known for 2 things. One is a recording studio where giants like the Rolling Stones used to come in to record records. The other thing is that it's a hot spot of species diversity for freshwater unionid mussels. Historically, over 80 different species of freshwater mussels were found in this one stretch of river, which is more biological diversity than any other place on Earth. So all of my high school science fair projects and my first scientific paper when I was in high school were on the Unionidae. These species are notable in part because of their unusual life cycle. The adults are benthic filter feeders. They settle down into the substrate, and they filter algae out of the water column, just like you would imagine for any other clam or mussel. Most of the species are dioecious, so they have separate male and female sexes. The females are fertilized internally, and a single female about the size of my hand can have about 1 million offspring in a single season. And she broods her offspring in her gills, and then eventually, as they mature, she releases them into the water column where they attach to the gills and fins of fish hosts, and they parasitize the fish and they suck their blood. Eventually, they mature enough and they drop off to develop into benthic filter feeding adults. On top of this, they have an unusual pattern of mitochondrial inheritance, where the males inherit their mitochondria from the paternal line, their biological fathers, and the females inherit mitochondria from their maternal line. So here we have these rapidly evolving phenotypes. We have parasite-host coevolutionary processes. We have this transition from herbivory to carnivory over their life cycle, and there's lots of interesting biology to study, but we didn't have genomes for these sequences when I started my faculty position. There have been some environmental changes in the region where there was a dam that was put in place in 1918. It was one of the first dams along the Tennessee River. And by 1924, natural historians who came over from Europe noted that there were species that used to be extremely common that had gone missing completely. On top of that, it is the most polluted stretch of river in the state of Alabama, which is a great thing if you grew up there. And so that pollution load that's very heavy has resulted in strong selection on these freshwater mussels, and now only 40 species of the original 80 remain. This isn't a pattern that's specific to this region. Across North America, over 70% of species in Unionidae are threatened, endangered, or extinct. So we have this unfortunate scenario, though, where there are some species who have succeeded in spite of these environmental challenges, and they have tens of millions of individuals just in one reservoir, and their range goes all the way across North America. We We have other species that are in extreme population contraction and potentially at risk of extinction, and then some that have not been seen in the wild in more than 10 years. So here we have this broad difference in effective population size, and most of our species come from this one specific region that has common environmental factors. So here we have this system that's seemingly a perfect fit for nearly neutral evolution. So when I got my faculty position and I got my startup, you get to blow it on any crazy idea you have. So I contacted Jeff Garner at the Alabama Department of Conservation. He's one of the world's experts on freshwater mussels and the co-author of this beautiful book. He shipped us freshwater mussels, and we did what we do. We threw them in the blender. We got DNA out of them. We got RNA out of them. We have new genome assemblies for 12 different species of bivalves. And my master's student just told me right before this that she's now got gene annotations for all of our species. So now we can start to do evolutionary genomics in this clade that's of conservation concern, where genomes weren't available before. We published the second genome for any unionid bivalve. Chase Smith has also done a lot of work, and Sophie Breton on Unionidae. So at first we got a genome. It was a lot harder than I anticipated. If you want to talk about challenges in getting long reads for Unionidae, I could talk for hours, but I won't. So we got our genome. I thought at first this would be a resource release. We would send out this to the field, and it would be something that people could use to make PCR primers. But because I just can't help myself, I took a quick look at the duplicate genes because that's what I study. You can use a molecular clock to plot the age distribution of duplicate genes, and you can fit birth-death models to the distribution of ages. And there were some gene families, as we did this analysis, that were so large, we could in fact fit separate birth-death models just for that one gene family. And when we did, we found significant differences between these gene families and the genome-wide background for some of these clades. When we did, we find gene family expansion that reflects the biology of the organism. We have these unusual mitochondrial management proteins, which fits with their dual uniparental inheritance. We have these anticoagulation proteins, which we think is important for their larval stage. We have these heat shock proteins, the cytochromes, and their interacting partners, the ABC transporters. We have these chitin metabolism genes that are important for the shells, and we even have these opsins, which we think are important for the ways that they sense light. So here, using this computational approach that's completely agnostic with respect to function, we recover some things that we didn't expect and some things that make sense in light of the biology of the organism. And we see signatures of selection with dn over ds greater than 1 for dozens of copies across this genome. So this was the first genome that we published, Madeline nervosa, but there were two species on our list that were very interesting. One is Elliptio spinosa. It's critically endangered and hasn't been seen alive in the wild since 2008. John Wares at the University of Georgia had DNA that was taken from one of the last individuals ever found in non-destructive sampling. We got a genome for it and for its counterpart with large population sizes, Elliptio crassidans. We repeat the same type of analysis, and we see selection on gene family expansion in some of the same categories. But there are a few differences. The opsins and these chitin metabolism proteins were non-significant, and we see much smaller effect sizes for some of the categories like these von Willebrand factors. Alternatively, there's another type of anticoagulation protein, the fibrinogens, that shows very strong selection. And that's seen both with the gene family amplification and with dn over ds analysis. When we look at our endangered species, we see smaller magnitude effects on several of the gene families. Several of them are not significant at all, and it's only a few of the categories that show strong selection. If we look at the number of duplicate gene copies that have signatures of selection with dn over ds greater than 1, then in every category except these fibrinogens, the species with the large population size has more copies with signatures of selection, which is exactly what you would predict under nearly neutral theory, that adaptation is more efficient in large populations compared to small populations. And if we look genome-wide, then we see a massive difference. And if you adjust for the background rates of gene duplication, 50% more copies have signatures of selection in the large population size species compared to the small population size species. And I ran into Mike Lynch recently, and I said, Mike, it looks like these freshwater mussels read your textbook. It fits exactly with all the predictions if you're looking at duplicate genes. There's another prediction from nearly neutral theory, and that's that transposable element insertions are largely bad, so they proliferate in small populations. When we look at our data, we actually observe the opposite. In orange, we have our small population size, in yellow, and it has the smallest amount of recent TE proliferation in the entire clade across all the species that we've looked at. And we compare it to the large population size individual, they have a half a gig of repeats across multiple families of TEs. That has resulted in a genome-wide expansion in repeats in the species with a large population size. We've been working on trying to find simulations to explain this, and right now I cannot find any neutral scenario where I can get anything similar to this result at all. So here the transposable element content is backwards of what you would expect from nearly neutral theory, which is also puzzling given the usual interplay that we see between transposable elements and duplicate genes. This is only the very recent TE expansions, not the stuff that's much older in the genome. All right, so this is the type of work that we've been doing on nearly neutral evolution for duplicate genes and transposable elements in large and small population sizes with our new reference genomes that we've gotten recently assembled and now annotated. We want to do similar types of analysis to identify gene families that are important for adaptation in these species of conservation concern, and then also to look at how these nearly neutral theories hold up across the entire clade, where we have multiple orders of magnitude for effective population sizes to see how TEs and duplicate genes behave. All right, and I want to thank several people, especially John Wares, who shared this specimen for Elliptio spinosa, which he got in 2008. I'm also grateful that he never cleared out his freezer. And also because he was underfunded at the time, he couldn't use a Qiagen kit and had to do a phenylchloroform extraction, and that is the only reason why we got the long reads for this genome, and it's one of our better genome assemblies. So a stroke of good luck. Yeah. Don't clean your freezers. Yeah. Do we have time for I have no timer up here. Okay. I'm either 5 minutes early or 5 minutes behind.
That was so cool.
Thanks.
So you saw less transposable elements in the small population size.
Correct.
I'm not familiar with the theory, but couldn't there be a theory that they're just being lost due to drift?
Generally speaking, the loss via drift is difficult on short time scales. And you get so much proliferation that getting enough deletions to remove them is a bit of a challenge. So the theory says that you have these repression mechanisms, and they're more likely to break and be tolerated in the population if the population size is small. So then that allows the Ts to proliferate and get the upper hand. And it's also harder to get a second repressor to come in if you have a small population size compared to a very large population size. Yeah, that's the theory, yes.
Following up, could they have been purged from really low population sizes? Basically, anything with too much Ts in them would have been died out, and then what you have left is some.
Right, what if they got purged from small populations because they're like recessive deleterious or something? We set up the simulations and the simulations say no. The other thing we looked at is perhaps you get less effective recombination. So then your TEs jump and they're detrimental, but they're next to something that's essential. And so you can't get rid of the TE by unlinking it because you don't have enough individuals to get the double recombinant. That's one hypothesis. We set up the simulations. I can't get it from that either. And I mean, so far I have not found an explanation, and we're kind of scratching our heads. One thing that could explain it is if there's a transposable element that copied something and hitchhiked on a beneficial mutation, like what we see in humans sometimes with ERVs and developmental genes, then that would make it impossible to shut off the TE on short time scales, and it would be proliferating, but you can't get rid of it because it's next to something beneficial. That's one possibility, but I'm not sure if that's the only possibility. Yeah. All right. Thank you so much.
Thank you so much, Rebekah. That was very nice. Our next speaker up is Erin Voss, who's a graduate student at the University of California in Berkeley, who's interested in evolutionary and conservation genomics of California rodents.
Awesome. All right, and that's what I'm going to talk about today. So hello, my name is Erin Voss, and I'm a grad student at UC Berkeley. And today I'm going to share some results from a chapter of my dissertation about genetic diversity across 3 species of endangered kangaroo rats. So the relationship between population size, genetic diversity, and conservation status is not always clear or predictable. For many years, we've observed that endangered species often have low genetic diversity as a result of their small population numbers. And relatedly, we sometimes observe phenotypic defects in endangered species, such as the kinked tail of the Florida panther. And as we just discussed, this is because deleterious mutations can rise in frequency due to the strength of genetic drift in small populations. However, some species seem to retain high levels of genetic diversity, and others, like the vaquita shown in the middle, appear to have purged deleterious variation from their populations. So today I'm going to explore this topic across 6 species of kangaroo rats with whole genome sequencing data that was funded by the California Conservation Genomics Project. So the California Conservation Genomics Project, or CCGP, is a big project that was funded by the California state government in 2019, and it's really the brainchild of Professor Brad Schaefer at UCLA. And the goal of the CCGP is to survey genetic diversity broadly across the state and inform conservation efforts going forward. And it's also a big collaboration among PIs from across the University of California system. So my advisor, Michael Nachman, and our lab are working on several projects funded by the CCGP that focus on California rodents. And today I'm going to talk about kangaroo rats, but we also have projects on voles, deer mice, and other rodent groups. And I've listed a couple of presentations by others in the lab who are going to talk about those. So first, I'll tell you a little bit about kangaroo rat biology and about the species that I work on. And then I'm going to take a step back and describe the questions that I plan to address. So kangaroo rats are rodents in the genus Dipodomys in the family Heteromyidae, and their closest relatives are gophers and beavers, so the term rat is not quite correct here. And they're only found in North America, and they're really iconic species across the deserts of the American Southwest. So they're uniquely adapted to live under dry conditions, and they can survive indefinitely without drinking any water. And they're also granivores. They gather seeds in their cheek pouches, which I'm showing in the close-up image, and cache them, and that really shapes the distribution of plants in the ecosystem around them. And this project is entirely museum specimen-based, but I have had the chance to catch kangaroo rats in the field as a first-year graduate student and then as a teaching assistant taking mammalogy, and they are just absolutely beautiful animals. Okay, so now I'm going to tell you about the 6 species that I focus on. And that I'll use for the rest of this talk. So there are 3 species of endangered kangaroo rats in California. So 2, the giant kangaroo rat, Dipodomys ingens, and the San Joaquin Valley kangaroo rat, Dipodomys nitrotoides, are endemic to the southern Central Valley, and 95% of their habitat has been converted to agriculture. And Dipodomys stevensi, Stevens kangaroo rat, occurs in Southern California in the region between Los Angeles and San Diego, and much of their habitat has also been lost to urban development. So I'll contrast these with 3 species that are not threatened. So at the top, Dipodomys hirmani and Dipodomys nitrotoides, panamontinus, both have moderate ranges, and Dipodomys miriami, Miriam's kangaroo rat, has a large range and is found from California, east into Texas and south into Mexico. And I'll use the colors on the map here to refer to the species throughout the rest of the talk. Okay, so with these kangaroo rats, I'm going to explore 3 questions. First, do the endangered species show reduced genetic diversity or increased runs of homozygosity compared with non-threatened species? Second, does genome-wide heterozygosity vary across species with different range sizes? And third, how does demographic history and past effective population size correlate with present genomic diversity? So to answer these questions, we first generated a new high-quality reference genome using PacBio for Dipodomys miriami. And this genome was assembled by Merle Escalona, who's an awesome genome scientist at UC Santa Cruz. And we also generated 10x Illumina whole genome resequencing data for 142 Dipodomys individuals that we mapped to our new genome. And again, all of these samples are museum specimens, and they came from the mammal collection at the Museum of Vertebrate Zoology at Berkeley. Okay, so here I'm showing a map with our sampling, and I want to highlight that we have 35 samples from endangered species, including 8 from Dipodomys ingens, 6 from Dipodomys stevensi, and 21 from Dipodomys nitrotoides. Our sampling for nitrotoides is particularly strong, with 11 samples from subspecies Brevinosus, 5 from nitrotoides, and 5 from subspecies Exilus, which is presumed extinct and hasn't been seen since 1992. So those last 5 samples are ear clips from outside of Fresno in 1989. And so they really provide a unique window into that population in the last few years of its existence. So first, I wanted to confirm that all species are distinct and monophyletic. So I used the whole genome resequencing data to perform a principal components analysis and build a phylogeny. And you can see the endangered species fall in the middle of the tree and are highlighted in red. And the other thing you can see is that these 2 analyses support each other with the 2 sets of sister taxa, Merriami and Nitrotoides at the top, and Panamontinus and Hirmani at the bottom, clustering together on the PCA. And you actually can't even really see the yellow Nitrotoides because it's overlapping so closely with Merriami. Okay, so now that I have described basically our sampling and evolutionary relationships among species, I'm going to turn to our conservation genomics questions. So first, do endangered species show reduced genetic diversity or increased runs of homozygosity? So we looked at heterozygosity in sliding windows across the genome. And here I'm showing the proportion of sites that are heterozygous summarized by subspecies, and that's why there are multiple bars for some of the species. And we observed quite a bit of variation in heterozygosity across endangered species. So Dipodomys nitrotoides in yellow has pretty high genetic diversity, while Dipodomys ingens and Stevensii have lower genetic diversity, but they still fall within the range that we observe in non-endangered species. And we didn't observe much evidence of runs of homozygosity in our data, which is encouraging because it suggests that inbreeding isn't a major concern for surviving populations of Dipodomys. The one exception to this is that we did observe runs of homozygosity in samples from Dipodomys nitrotoides exilis, which is the extinct subspecies. And here I'm highlighting 2 scaffolds that show long runs of homozygosity in this individual. So despite those high background levels of heterozygosity that we observe in nitrotoides, exilis does show signs of inbreeding prior to extinction. So second, I wanted to know, does genome-wide heterozygosity vary across species with different range sizes? So sometimes we use range size as a proxy for census population size, and we might expect that species with larger ranges and larger census population sizes have higher genetic diversity. However, as I'm showing here, we don't really see a correlation between range size and heterozygosity. Okay, so since I didn't learn much about genetic diversity from current species ranges, I turned to demographic inference to explore ancestral effective population size and ask how demographic history might be shaping current genetic diversity. So I estimated past effective population size for each species with pairwise sequentially Markovian coalescent analysis, or PSMC, which estimates past population size based on local recombination and coalescent events across a single genome. So here I'm showing the PSMC results for Dipodomys merriami, the widespread species. And PSMC is known to be less reliable for modern timescales, so we tend to take the first few time bins with a grain of salt. But looking past that, you can see that merriami appears to have undergone a decline during the last glacial maximum and then expanded again in recent years. Okay, and here I'm showing the PSMC results for every species. And the specific details aren't super important here, but there is quite a bit of variation. And to explore the relationship between demographic history and heterozygosity, I took the average effective population size starting from 200,000 years ago up to 10,000 years ago for each species. And that's shown with the bracket above the plot. And I did find a strong correlation between historic effective population size and present heterozygosity. So for example, in magenta, Dipodomys ingens has had a small and decreasing effective population size for the last 1 million years, which might explain why we see lower genetic diversity in them than Dipodomys nitrotoides in yellow, which has historically had a larger population. So to summarize, we did not observe a reduction in genetic variation or runs of homozygosity in endangered species, except in the extinct Dipodomys nitrotoides exilis. And we did observe quite a bit of variation in genetic diversity across species, but it was not correlated with present range size. Instead, demographic history and past effective population size seems to be a better predictor of current genetic diversity. So to return to my initial framing of this talk, we saw a range of genetic diversity in endangered species, with Dipodomys stevensii and ingens showing low to intermediate genetic diversity, and Dipodomys nitrotoides showing high genetic diversity. And we also observed a non-endangered species, Dipodomys panamentinus, with low genetic diversity. So today, I've presented results regarding neutral genetic variation, but I also have the data to explore deleterious mutations and genetic load. And that's my planned next step to wrap up this project. So I think it's an open question. I'm not really sure what I'm going to find, but I'm looking forward to exploring that. So ultimately, I came away from this work feeling that prospects for kangaroo rats in California are somewhat mixed. Our results suggest that genetic diversity will not be an impediment to survival, particularly for Dipodomys nitrotoides, which appears to be genetically diverse and has the potential for resilience. And I'd also like to note that Dipodomys stevensii was recently downlisted from endangered to threatened due to successful conservation efforts in Southern California. However, we have to remember that 95% of these species ranges has already been converted to human use. And I think if that continues, we will eventually start to see some breakdown of the genetic diversity across the landscape. And that is all I have to share with you today. And I'd like to thank all of my co-authors because this was really a big group effort to generate and analyze the data, and all the people at Berkeley that have supported me, and thank you all for listening. If I have time, I'd like to take any questions.
I'm wondering how well can kangaroo rats dispose of habitats?
Not well. Yeah, so a big issue, and one that I can't really address with this data, is that fragmentation is kind of like really preventing populations from connecting with each other. But our data is sort of more across the landscape, so hopefully you can figure that out with future work. Yeah. Mm-hmm.
How well does range size actually correspond to population size?
Yeah, that's a good question. For the kangaroo rats, I'm not sure. Yeah. Stay tuned. Yeah.
So in relation to his question, if the populations are isolated and you are pulling all the information from the different populations and you get a huge estimation or a large estimation of genetic diversity, but the individual populations are really endangered.
I think that that is definitely a possibility. For some of the endangered species, because the ranges are so small, we do actually have we'll have sampling from one population that will be good. And then we just will have missing data from the other populations. And so I feel like we can address genetic diversity within a population. It's just that we can't address fragmentation from other populations. Does that make sense? Yeah.
Thanks, that was super interesting. So your PSMC plots go back about 1 million years.
Yes.
Which is pretty typical of PSMC plots. But how old actually do you think these species are?
I believe the most recent split was at least 3 million years ago for the more closely related ones, and the whole genus goes back to about 12 million years.
Okay.
All right, great, thank you guys.
So our next speaker is Andrew Black, who's a senior computational scientist at the University of Oregon, who's interested... Oh gosh, sorry. Oregon State University, my bad. Who's interested in thinking about mechanisms at the intersection of conservation and evolutionary genomics in natural vertebrate populations.
All right, thanks for the introduction. Again, my name's Andrew Black. I'm joining you from Oregon State University. The great thing about genomics is it's highly portable across taxa. So these are a couple of the ongoing projects I'm working with, everything from lemur, who I think the next talk is on lemur, to lesser prairie chicken. We actually just got our paper published on lesser prairie chicken genomics, which is super exciting. But today I'm going to be focusing on one of these projects, and that's the white sands pupfish, or Saprododon tularosa. They're not as charismatic as kangaroo rats, but I think they're a pretty cool species, weird, chunky little fish that occurs in desert springs. So This work was just published in the National Academy of Sciences in May, so just in time for the Evolution Conference, which was a big bonus. It's entitled Rapid Vertebrate Speciation via Isolation, Bottlenecks, and Drift. I do want to note that this is very much a group effort with the authors in bold having equal contributions to this project. So the white sands pupfish is a small desert killifish that's restricted to the Tularosa Basin in New Mexico. They were designated as endangered by the ICN in 2022, and they're currently managed as 2 evolutionarily significant units, or ESUs. The ESU designation is due to considerable differences at several allozyme and microsatellite markers, as well as key differences in salinity and water flow, morphologic and merisitic characteristics, as well as very different parasite communities. So this provides a great backdrop to explore patterns of divergent selection between these really cool isolated desert springs. So that was our primary objective going into the study. I'm not going to get into the methods too much with this talk, but we obtained samples from 3 different populations, as you can see here on the map. Salt Creek, Lost River, and Malpaya Spring. The 2 different colors signify which ESU they belong to. So I'll be using blue and black throughout the talk to signify which ESU these populations belong to. For each population, we use 2 different genomic approaches, so whole genome resequencing and pool-seq, 15 to 30 samples from each population. So we first reconstructed the demography using the joint site frequency spectrum. So you can see my mouse. On the y-axis, we have effective population size, and then we have time on the x-axis from past to present. The first thing I want to signify here is from the ancestral population. You can see about 95% of the ancestral variation was retained by the Malpaya Spring ESU, whereas only 5% was retained by the progenitor of the Salt Creek ESU. But you can see that there were 2 very drastic bottleneck effects after population expansion. 2 dates I want to point out, determined by the most likely model. First is the dating of that initial split, which was about 4,000 years ago. This is consistent with Stockwell et al., a 2013 paper, and coincides with a vicariant lava flow event that's geologically dated to have transpired 5,000 years ago. So this was the primary force that split the ancestral population into the 2 progenitor ESUs, if you will. Secondly, you can see here that the model determined that Salt Creek and Lost River, both part of the same ESU, diverged 59 years ago. So we know from the historical evidence that humans actually pulled about 30 fish from Salt Creek to found the Lost River population that's occurred 48 years ago. So it's a good kind of quality check with the model. So, as we all know the importance of genetic diversity in conservation, we then estimated individual heterozygosity for all of our whole genome resequencing samples. You can see here that there are significant differences both between populations as well as ESUs. Malpaya Spring ESU had the highest relative amounts, and Lost River had the lowest. However, if you look at the overall levels of genomic diversity, they're really quite low. So this is from a previous paper. The white sands pupfish is here. These are heterozygosity estimates compared to closely related species, so other cyprinodontiforms. And you can see it's on the low end of the spectrum compared to the other ones, down near those that are either selving or inbred laboratory lines. So definitely a conservation concern. So we then wanted to calculate the FST, so mean global levels of genetic differentiation, both within and between ESUs. So below the diagonal here, we have the whole genome resequencing comparisons, and then above the diagonal, we have the pool-seq. So first want to draw your attention to the comparisons in the populations within the Salt Creek ESU. Again, these were only separated for about 50 years. Very high levels considering the very short amount of time they're separated. So 0.04 to 0.14. However, we're totally blown away when we looked at the global FST values between ESUs. And we're looking at values of 0.2 to 0.4. So again, very, very high levels. We went back and double checked and triple checked our data, but it was consistent. So we thought going into this that we'd find something like this. This is not our study. This is from Hess et al. In 2020, but where you see a pattern of these islands of differentiation, right? You have these peaks that kind of stick out from the background distribution that are due to things like selective sweeps. So this is the pattern we were expecting to see with our genomic data. We see something very different, right? So definitely not replete with the classic islands of differentiation, either the pool-seq data or the whole genome resequencing. We have a very high background level. However, we still wanted to see if we could identify some genes involved with this divergence between these two ESUs. So we searched the genome for a list of candidate genes involved with osmoregulation, aquaporins, transporters of ions, things like that. We only found 2 genes in the upper FST distribution. However, when we looked at these regions, we found really no evidence of selective sweeps. So that is things like heterozygosity, Tojima's D, DXY, not a clear pattern. So at this point in the study, we really shifted from like an intraspecific comparison of these two populations. We're like, maybe we're working with 2 different species. So we kind of shifted our project goals, wrong way. And these are 2 phylogenetic trees, one using nuclear markers on the left here, and then we also reconstructed the full mitochondrial genomes for our samples. We used the sheep's head minnow as an outgroup, and you can see both of them are very clearly reciprocally monophyletic. So good indication of distinct species. We also then continued on to perform some actual species delimitation tests using BPP. Don't expect you to read all this. Just want to point out that we use both nuclear and mitochondrial loci, a couple different algorithms, and provide very strong support for these 2 distinct species. Then finally, we calculated DXY, so absolute levels of divergence between each of these populations, and also included 2 recognized and known species of pupfish. And you can see here, these are the comparisons between the 2 ESUs in the red box, and they're very similar, if not slightly higher, than those between 2 recognized species of pupfish. So perhaps I've been spending too much time with my 9-year-old son, but I think Dr. Seuss says it best. We walked into this with 1 fish or 1 species, and we walk out of this project with 2 fish or 2 species. So our general conclusions are that this is really transpired due to the very small effective population size in these populations, as well as the massive bottlenecks that we modeled. This has really led to a new vertebrate speciation event just within 4,000 to 5,000 years. So because the Malpaya Spring ESU kind of retains the majority of the variation in our data set, we're kind of recommending that it takes a new species name. We're picking enchanted pupfish for 2 reasons. One, for the kind of the lure of the drift speciation event, and then 2, because the species found in New Mexico, which is the land of enchantment. The next step's really getting in there and doing a formal taxonomic assessment of the species, which is hard. It's on a US Air Force base, so you can't really just easily walk on site. But that's kind of the next step. It's also important that a refuge population should be established or maintained for the Malpaya Spring population, just as a hedge against extirpation. Yeah. So with that, I'd like to acknowledge a lot of collaborators, as well as funding, and take any questions.
normally rule out any selection pressures being involved in the divergence?
We can. We just couldn't really, because of all the high levels of drift that we're seeing in that system, it's hard to identify selective signals there. They could be there, they're just hard to uncover. But especially with the small effective population size, coupled with the high levels of drift, it makes it very difficult, I think, in that system. Yeah, so these two species differ drastically in their morphology, significantly, as meristics as well. Yeah, Ruth.
I was looking at the phylogeny, so it looks like the Malpais population evolved from the other population. Is that how you think the divergence happened? So they sort of split and speciated from there?
Well, I mean, we know that there is the initial split, at least according to the model, right, where we have ancestral population, and most of the variation went towards Malpais spring ESU. There's very little that actually went to the other species or ESU. But the actual timing, we haven't done any timing on that. Good question, though.
Sorry if you said this and I missed it. What drove the decision to do pool seq as opposed to individual whole genome sequencing?
I think, I don't know. I started as a postdoc with this, and we had the data in hand. So beyond that, I think it was just, it's also a nice kind of validation, having two different approaches and getting the same results. But yeah, I'll stand that data. That's the short answer to that. Yeah.
Can you wildly speculate on, in your opinion, can you see reproductive isolation?
Yeah, well, actually, I don't need to speculate too much on that. There has been studies where they're able to take a fish from the two different ESUs, and they do produce viable offspring. So they are able to reproduce together, which I know where you're going with that question. But a lot of pupfish species that are recognized produce hybrid swarms in nature. So even though they produce viable offspring, we don't determine that as ruling out that these should be two species. Yeah.
Another question of wild speculation. How common a mode of speciation do you imagine this could be? Is this sort of thing happening all the time?
I don't know. I think, of course, that's where my mind goes, especially, starting looking at other pupfish species and seeing if they have similar mechanisms. But how common and prevalent this is, I think it's definitely worth paying attention to and looking into. Yeah, thank you.